G'day guys, this is Supplemental on Leucine. A few things that I need to sort of cover. There will be cases where you may show this to other people, my previous video, and people may say, well, there's evidence showing that leucine is associated with certain poor outcomes or whatever. They'll use this sort of nonsense and all that. Pretty much that's all epidemiological. It's associated data. They haven't demonstrated a mechanism. See, what I showed in the previous video, I showed mechanism. That's important. What mechanisms work? What is affected? Which pathways are affected? That's the important part. You can design any study to show an association with something. Give you an example. You can be on a standard diet. That means going to McDonald's, eating French fries, all the different sort of stuff, a lot of sugar, a lot of fat at the same time, engaging the Randall cycle. So rather than having an insulin to glucagon ratio of 1.3, you can be up to 70. See the difference? Now, you can actually put the same amount of leucine in there, but you're going to get a completely different effect, one from the other. The study that I showed were rats on a standard chow, so the standard species-appropriate food for a rat. A species-appropriate food for a rat is more high carbohydrate, very little fat. So that's the diet for rats. It's not our diet. It's not diet of other animals, specifically for rats. So rats, while are classified as omnivores, mostly consume a small amount of fat and mostly consume far higher amounts of pretty much, because I'll eat certain insects and stuff like that. And a lot of those things are high in protein, but very much lower in fat. A lot of the insects and things like that. So, and grains and stuff like that. Those are the things, or plants, certain types of plants and stuff like that. That's what they'll consume. They don't consume, filet mignon, as an example, in the wild. That's not what they consume. And so when you actually look at, their species-appropriate diet, which is what they were doing on the previous one, there you don't see any of the inflammatory markers. Everything's, at a baseline. And then you give an intervention, so leucine or whatever else, taurine or whatever else, and then you see a response, a dose response. You take away that thing, you see a reduction in that response. You increase that thing, you see an increase in that response. That complies with the Bradford Hill criteria. You don't see an inverse relationship. That means you continue giving, so you give another group of rats and they show the complete opposite on the same diet, the standard chow. Now, there is what is called obesogenic chow, and that is a chow that's trying to mimic the SAD diet, and so it's full of seed oils, multidextrin, for the sugar, and all these other wonderful concoctions which they throw in. They usually will use, sometimes they'll use milk protein like casein, or sometimes they'll use a bovine one, or they'll use some other source. It can vary depending on the study, but generally speaking, they're on a chow that's primarily Randall cycle activating. So we're talking about a smaller amount, less protein and more fat and sugar. So, which engages the Randall cycle and actually causes inflammation, obviously. So under those sort of circumstances, completely different, because under those sort of circumstances, you've already activated mTOR in the fat cells very high. You've already activated the sort of the other pathways, IGF-1, insulin, all these high. And so when you throw in leucine, what it does is it actually amplifies the signal. So it can actually make things slightly worse. That doesn't mean it's leucine that's causing it. It's the actual diet in combination. And that's what we need to always keep in mind, that, you know, having a meat patty with all the other crap can potentially push your insulin to glucagon between a 70 to 1-fold. That's massive. Where if you just eat a steak, your insulin to glucagon ratio is 1.3, which is quite low. Because remember, fasting is 0.8. That's fasting. So it's just slightly, you know, it's not, you know, it's a bit over fasting at 1.3, but you still got glucagon in the system, and you've got very much lower levels of insulin compared to any different other state. So let's say you do a potato diet, and we know people can lose weight on a potato diet, a pure starch diet. They're not engaging the Randle cycle. The insulin to glucagon ratio is 4 to 1. Got it? So it's still much higher than a steak. But it's not to the level where it's going to cause derangements. But if you're having a burger with French fries, high fructose corn syrup drink, and whatever else crap, you know, orange juice and all these sort of things, you throw all those things, that is what it is classified as an obesogenic. Now, somebody can basically consume, let's say they're having all the same crap, but they decide, no, they're going to have a vegan burger, which is made from plants, carbohydrates, stuff like that, you know, veggie burger or whatever. Well, the amount of protein and fat is going to be slightly reduced. So their insulin to glucagon ratio is going to be different. It's going to be slightly lower, and their inflammation is going to be slightly lower. Their Randle cycle activation is going to be slightly lower compared to a SAD dieter. That's why they always compare the vegans and the vegetarians with the SAD dieters. Not that they're actually any healthier in a massive way. They're just slightly better, because even though they're eating this junk food, they're engaging the Randle cycle at a lower gradient because they've got less fat and there's more sugar. And when you've got more of one of the, the substrates that we use to produce ATP, then it's going to be less of any, the Randle cycle activation is going to be at a much lower gradient. And that is the difference. And we know, if somebody's on, on a low fat or low carb diet, then it's much harder to engage the Randle cycle under those circumstances, So you have to really work at it to try and get, most people will get the satiety signals. That's not going to happen. And that is why we see the difference. The only difference is that one is nutrient deficient, which is the low-fat diet, and you will pay for it. Most people can't sustain it beyond 5 years. Some do by cheating and a whole lot of other things, but that's a different question altogether. The issue here is that we need to understand the nuancing about these. When we look at these studies, look at the design. What are they using? Are they using the high-fat chow? If they're using that, forget it. It's a Randle cycle issue in the rat. It's garbage. You cannot basically you throw in extra leucine, you're actually throwing more fuel to the fire. You're actually putting in more elements to amplify the insulin to glucagon ratio and the and the anabolic signaling. So yeah, they're already high. You're pushing them slightly higher. So you will see slight differences in worse outcomes because of a higher Randall cycle as well activation. So that's how you have to think about these things. And the thing is, what does Bradford Hill, which all these epidemiologists quote but ignore, he actually says it quite clear. You can't have an inverse relationship. Something that actually causes something must always associate. What I showed you yesterday is something that doesn't associate, does the complete opposite. But obviously, if you were to throw in there a whole lot of seed oils and refined sugars and all that, you'd get a very different outcome, even in the rats. And that's really important. Let's not forget, it's well known. Epidemiologists have recorded it, that the rats in New York are very sick and very fat. I wonder why, because they're eating the same crap as most people eating, the fast food crap, which is Randall cycle activating obesogenic. It's exactly like the same crap that they give, the obesogenic high fat, as they call it, which is really high fat, high sugar. It's not, a high fat ketogenic or low carb style of diet. So we need to understand also what they mean when they're talking about, because in their paradigm, in their thinking, they're comparing between a diet that's got a lot of sugar and fat, which is a standard SAD diet, and a diet which is low fat. That's what the two that they're comparing for, between vegetarian, vegan, plant-based, and a SAD diet. That's the comparison. One is less activating in terms of the Randall cycle, and the other is more activating. That's the difference. But obviously, they don't provide that nuancing. That's what I'm providing you, so you can actually, when you read any study and somebody throws at a study at you, you go through it and go and look and go, okay, they're activating the Randall cycle in this animal model or in this human group. Busted. You don't have to, beyond that, it's all just nonsense. If you're going to look at something, you have to put people on a non-inflammatory, species-appropriate diet and then add interventions and see what the results are going to be. That's the difference. And this is why there is an association between leucine, and there's a couple of cancers out there that they've looked at. Again, these are people that are unhealthy and people that are engaging the Randall cycle, that are on a SAD diet already and still on a SAD diet, even though they have cancer. So when they do give them leucine, they do notice an amplification of the Randall cycle. Well, they don't understand that, but they actually notice slightly increasing inflammation. Now, they haven't proven a cause and effect here. It's an association, and they're looking and they're extrapolating from the increase due to the Randall cycle, increase inflammation markers, and they go, oh, it's actually doing bad things. But just because when you do actually amplify the Randall cycle effect, it actually makes the outcome, the cancer outcome, worse, because you're pushing a lot of the anabolic pathways, which is the last thing you want to do when you've got cancer. That's the problem, And so there is a difference between being at one state of insulin to glucagon of 1.3 on a carnivore diet, and a difference being on a Randall cycle, high-fat, high-sugar, SAD diet, and being at 70, an insulin of 70 to 1. We're talking about here the low fat is just, at 4 to 1. So even the low fat is much better than a potato diet. And then bang, in terms of Randall cycle, and then bang, the SAD diet up here. And just because the SAD diet compared to the potato diet or the vegan diet or whatever is worse because of the Randall cycle, they don't understand that. They go, oh, It's not the sugar, it's not the oils, it's nothing, none of all this other crap, the high deuterium foods and all this. No, it's the little meat patty, the healthiest part of the actual meal. Of that gets accused. So yeah, it's a play on things, and we need to understand that is what we're being conned, whether due to ignorance or because they really know and they're actually working for the food industry and getting well paid to promote certain agendas. So keep that in mind. Now let me just share my screen. So Bradford Hill criteria. Sir Austin Bradford Hill, who is an epidemiologist who came up with this. The definition, there are 9 criteria. So strength. A small association does not mean there is not a causal effect. Through the larger the association, the more likely that it is causal. That means we can infer a causality. Infer. This is Wikipedia, so I couldn't actually find the other one. So we'll have to bear with this. It's an inference in an association. That means like the inference between odds ratio of 10 to 30 with an average of 20 odds ratio, which is thousands of percent, is what was related to tobacco smoking and cancer, lung cancer. Anyway, consistency, the ability to reproduce, reproducibility, consistently finding observed by different persons in different places with different sample strengths and likelihood of an effect. So they're always finding the ability to consistently reproduce similar results. Well, we know, because the stuff that I showed you, the mechanistic study, showing exact which mechanisms are involved, it's not consistent with the other literature. And the other literature is piss poor associated crap. So it's already failed. All the other studies have already failed because mechanistic studies are different. So that experimental mechanistic studies have actually already invalidated all those others that accuse leucine of being problematic, because there is you can't always reproduce that effect. So no. Give me a bit of another go at it. Specificity. Causation is likely if there is very specific populations as a specific site and disease with no other likely explanation. The more specific an association between a factor and an effect, the bigger the probability of a causal relationship. That sort of more applies to viruses and bacterial infections and stuff like that. Temporality. The effect has to occur after the cause. And if there is an expected delay between cause and expected effect, then the effect must occur after that delay. Biological gradients, dose-response relationship. So greater exposure to something should generally lead to greater incidence of the effect. However, in some cases, the mere presence of the factor can trigger the effect. In other cases, an inverse proportion is observed. Greater exposure leads to lower incidences. So you've got to see some sort of, when you look at leucine, it's all over the place. There isn't that dose response. You can actually have high levels of leucine supplementation and see one effect, and high and low seeing different effects depending on which model it's actually put in, which means it's just associated, and it may be interacting with something. As I said, Randall cycle, the different sort of growth factors that it's going to interact within a Randall cycle state. So plausibility, a plausible mechanism between cause and effect is helpful, but Hill noted the knowledge of mechanism is limited by current knowledge, and that's true. There is, but he does actually put plausibility. So you've got to be the strength of the strength of a claim of causality must have plausibility. You must be able to demonstrate that some sort of mechanism. Now you can infer at a speculative level when you don't have the mechanism, but all the other things have to also comply. So all these things must be valid in order for the Bradford Hill criteria to be valid for that case. Coherence between epidemiological and laboratory findings increase the likelihood of an effect. So this is where we say experiments in the lab. Okay. However, Hill noted that lack of such laboratory evidence cannot nullify the epidemiological effect of an association. So if the association is extremely strong and it's dose dependent and all the other stuff is compliant, then he can say, well, we haven't done the laboratory work, but the likelihood that it's something else is unlikely because it's, if we discount everything else, the likelihood is that it is this causing it. We just haven't got, because in some cases you may not have the technology to be able to do it. Today it's much easier. So just keep that in mind is what he's talking about. But, obviously if you do have laboratory work like what I showed yesterday, the laboratory work showed, which is mechanistic, experimental, done in a lab, showing a complete inverse relationship, a complete different effect of leucine supplementation, which completely blows out of the water all the actual other studies. Because remember, none of the studies that are done are done when it comes to different nutrients and stuff like that, and different they're all associated, and usually the majority, overwhelming majority, tend to be animal research. It's because of the ethics. You can't experiment on people with cancer. It's just one of those things. So all we've got is, these very poor questionnaire-based epidemiological stuff of, what do you eat? Which is pretty much junk science, because it's just basically very poorly captured. And usually the actual odds ratio is really poor. It's less than 2. I mean, if you look at most epidemiology on meat, even processed meat, it's about, I think the highest I've actually ever seen is about 27%. 27% is nothing. It's statistical noise. It's an odds ratio of 1.27. That's what it is. In order to have, in epidemiology, even the basis for to conduct to put forward, that's the right word, to put forward an hypothesis, you need to have an odds ratio of 2 or more. And in order to infer that something could be causal, you need to have an odds ratio between 6 and 10. And if you have an odds ratio of over 10, the likelihood that it's something else, which is what we looked at before, the likelihood that it's something else is unlikely. So it's more than likely that is the plausible thing that's actually causing it. So the inference is much stronger, because we never can prove through association causation. All we can at very high odds ratios, like thousands of %, the likelihood it's anything else is unlikely. And that's how we were able to determine that tobacco smoking will, is something that will cause lung cancer. Now, it may not cause it everyone, but it still has an odd ratio, of 10 to 30. So heavy smokers, 30, lower people that smoke less, 10. And we notice less cancer in people that smoke less and more cancer in people that smoke more, and other health issues as well. So there is, a dose relationship as well. So it's pretty good, that sort of stuff. Now, experiment. Occasionally it's possible to appeal to experimental evidence, which is what I showed you guys yesterday. Analogy. The use of analogies or similarities between observed associations and any other association. That means with other components, it seems to another association seems to validate another sort of association as well. So there are similarities which one seems to reinforce the other, sort of thing. Now, in order to basically say that there is an inference, this is the important part, an inference, all these things have to be valid, all 9. Some authors consider also reversibility. If the cause is deleted, then the effect should disappear as well. So that's, so if you take away something, it should go away, the problem. That's a problem because it depends what it is. Like if you take away tobacco, if you've already got underlying damage, will it, and you've got a poor diet, in general, reversibility maybe under certain circumstances, but it's one of those really hard things. And it seems that some people have thrown that in to muddy the waters. So it's not something that I would use because, that's not always the case. Some diseases can take like decades to develop, so, it's reversibility is a problem, is problematic. Under certain circumstances, yes, we do notice reversibility when people go on low-carb diets or even vegan diets, disengage the Randall cycle, and we do notice improvements in a whole lot of biomarkers. So we do notice reversibility in that regard. But if somebody's too damaged, we don't. And then you need other biohacks and other interventions to sort of try and prevent that damage. Like, let's say somebody's too damaged in terms of cancer, or too damaged in terms of cardiovascular disease, then you have to do other interventions because you may have mitochondrial dysfunction. So even though you take away the stuff, you're not going to basically see reversibility because you have mitochondrial dysfunction. You're fermenting. You've got a lot of senescent cells, and possibly even cells that are cancerous. So things are really deranged. So taking away something isn't going to cause complete reversibility. So there is a bit of a problem with that some authors are pushing. So it has to be, if it was ever to be used like a 10th thing to be added, it would have to be nuanced, and it would have to have a criteria to it in order to basically, otherwise it just would make a mess of the whole Bradford Hill criteria in that regard. Obviously, this is was an attempt by Bradford Hill to try and come up with a solution to the problem of being able to infer causality or show a causal relationship between associated data. And remember, he was looking at not highly confounded data. This is a method of looking at low confounded data. Because remember, he was an epidemiologist. He wasn't a nutritional epidemiologist. This is not suitable for food, this whole method. This is suitable when you're looking at like one component is great. So if we've got two groups on identical on their species appropriate diet, and then we throw other things in and see what happens, this can be utilized for that. And this is why the Bradford Hill criteria was developed for, smoking, has been used for diseases like, viruses and things like, exposure, like poor sanitation. And, and you'll actually see that greater exposure should generally lead to greater incidence of the effect, So there's that sort of stuff. Where was the other one? And the point that was made in the beginning, for example, he demonstrated the connection between cigarette smoking and lung cancer. And this is what it was, the criteria came out of that research. So here we're looking at one product, one item, and looking at these sort of effects. So keep that in mind, that Bradford Hill criteria is specifically to that and doesn't really work well with diets, because there are the Randall cycle activation, there is the hormonal system, there's all these other things that are at play, and nutritional confounding is a big problem to be able to pull it apart. And so that's why if we really want to research nutrition, we should do experiments, or at the least uncontrolled randomized control sort of studies, which that means you don't lock people up, they're a free living, but you've applied a randomization in terms of what diet they're on. Now, it's very hard to determine compliance because they're not in a lab, so it's a much weaker thing, but it's way better than the current stuff, the current epidemiological claptrap that's out there. And there's a lot of it that's out there. And it's just nonsense, completely and utterly nonsense in that regard. So we can scrap all that. Now, with leucine, so leucine supplementation, a novel strategy for modulating lipid metabolism and energy homeostasis. So lipid metabolism is an important and complex biochemical process involved in the storage of energy and maintenance of normal biological function. Leucine, a branched-chain amino acid, has anti-obesity effects on glucose tolerance, lipid metabolism, and insulin sensitivity. Leucine also modulates mitochondrial dysfunction. We've covered that yesterday, representing a new strategy to target aging, neurodegenerative disease, obesity, diabetes, and cardiovascular disease. All through various studies have been carried out, much uncertainty still exists, and further studies are required to fully elucidate, understand the relationship between leucine and lipid metabolism. Yes, true. This review, because this is a review, offers an up-to-date report on leucine as a key role in both lipid metabolism, energy homeostasis in vivo, which is in the body, and in vitro in a petri dish by acceleration of fatty acid oxidation, lipolysis, activation of adenosine 5 monophosphate activated protein kinase, which is AMPK. You've probably heard it when you're in a fasted state, AMPK goes up. Okay, so leucine, when you're doing fatty acid oxidation, can actually amplify the effect of AMPK. So if you're on a low carb diet and you're actually consuming more leucine because you're on a carnivore diet, you can increase your fatty acid oxidation. Obviously, how much will depend on how much mitochondrial, if you're new to the carnivore diet, you've still got a lot of mitochondrial dysfunction and problems and all that. It's going to take some time to heal, but once you've healed, you should actually, get much better fatty acid oxidation. Now, somebody who's young, they will notice it. And I've known a number of people that are young, that I've got them on a carnivore diet, and they just rip body fat like that off because they have less mitochondrial damage. Us middle-aged people that have done a lot of damage with seed oils and other crap like that, and refined sugars, high in deuterium that damaged our mitochondria, we're still healing in that regard. So it also silent information regulator of transcription 1, which is the SIRT1. People are well aware of SIRT1. So even out of the NAD, you get PARPs, SIRT1, and all that, and that then creates nicotinamide through the salvage pathway. So leucine plays a role there as well in that regard. And we've actually covered proliferative activator receptor G co-activator 1A, which is PGC1 alpha pathways. We've covered that yesterday. Synthesis or secretion of adipokines, stability of the gut microbiome, very important. See, this is why people, when they're on a species-appropriate diet, eating foods that are high in leucine, carnivore, they actually see improvements in gut stability. Obviously, you still need taurine, which is animal foods, plus vitamin D to tighten the junctions, but also leucine plays a very important gut stabilizing role. Now, adipokines is things like leptin, things like that. I can't remember all of them. Adiponectin, I think is the other one. Just give you a sec. Okay. Adipokines, also called adipocytokines, that's true, are cell signaling molecules. Let's just increase that. Cell signaling cytokines produced by the adipose tissue, the fat cells, that play a functional role in energy metabolic status of the body, inflammation, obesity, etc. Notable examples of adipokines include leptin, so here you go, and this is probably one reason why I actually noticed myself when I spike deliberately some of my food with more leucine, I notice I can actually get like a fuller effect quicker. So I can go, I can actually blunt my hunger. And it's probably I'm deliberately pushing up leptin to signal and say, so the adiponectin, so is well known as a homeostatic factor for regulating glucose levels, lipid metabolism, and insulin sensitivity through its anti-inflammatory, anti-fibrotic, Yes, I know a lot of you oldies like to be on an anti-fibrotic diet and antioxidant effects. The next one's one of my favorites. And resistin is a cysteine. Again, you got to eat cysteine to be able to furnish the material, the amino acids, to be able to produce these things. They don't produce them themselves without providing the food that are needed. Rich hormones secreted from white adipose adipocytes, so white fat cells. They don't, you're not going to get it from brown adipose tissue, okay? Is involved in insulin resistance and links to obesity, diabetes, involved inflammation. Humans, resistin is expressed in white adipose tissue in rodents. Where are we? This one. Interleukin-6. Now, I get people that are on cancer to actually increase their interleukin-6. There are some peptides that can actually do that. And the reason for that is because you're actually increasing your T cells. And you need T cells to kill off cancer cells. So we're actually tricking the body, the immune system, to go, oh, there's a problem, to increase the amount of T cells. Got it? So it's a bit of a trick. I haven't made the video yet, but I will cover it. I have provided the information to some people that have approached me with their cancer situation, but there will be a video that will cover the peptides that are involved in it. Anyway, so many videos to make. So, as they usually say, good or bad, potentially useful as a marker of immune system activation. Exactly. Because that's what I want. Interleukin-6 can be elevated with inflammation, infection, autoimmune disorders, cardiovascular disease, and some cancers. That's true. But I like actually increasing it slightly more because I want to force the body to produce and put out a few more extra T cells to go after the cancers in particular. So just another biohack, so to speak. And tissue necrosis factor. Anyway, that's well known. We don't need to go any further that. So pretty much covers that. I'm not going to go through this whole review. People can go through it themselves. It's not the purpose of this video. Otherwise, it'll end up as a long supplemental. But that's normal with me. You should know that by now. Anyway, to date, leucine, as well as its metabolites, have been shown to possess promising role in lipid and energy metabolism by directly or indirectly increasing the oxidation of fatty acids and improving metabolic health. Leucine supplementation results in beneficial effects in the adipose tissue and skeletal muscles and intestines. Furthermore, leucine may have an additive effect and may alleviate mitochondrial dysfunction. Well, we showed that yesterday. It definitely does. Representing a new therapeutic approach against aging, neurodegenerative diseases, obesity, diabetes, cardiovascular disease. From a standpoint of a practitioner, leucine has the potential to be used as a functional additive for both human health and production of livestock. Yeah, I mean, that's true. I think a number of farmers and veterinary people are using leucine now to improve healing of animals. However, it is necessary to carry out additional experiments to elucidate its regulatory mechanisms. I actually had somebody leave me a message today on the video that I just made, and they pretty much confirmed what I know quite well. They said, Harry, thank you very much. I've been on taking taurine and my mental health stuff has improved, all these other things he mentioned. And then he mentioned that he was also doing leucine as well recently, and he noticed his pain in his knee and other things had actually, things had improved. Because the combo of the two, I've talked about it a number of times, just haven't made the video, but the combo is that, you know, taurine actually also helps with bones. Because what it does is when you taurine's, you know, as I said, it's a master regulator, but also can, in a state of fasting, if you take taurine, you amplify AMPK, fatty acid oxidation. Got it? And it will not raise insulin. But if you are taking it with leucine, it does the complete opposite. It amplifies the effects of leucine. It raises in the musculoskeletal, bones and muscles, it raises IGF-1, insulin, and mTOR, and also lowers myostatin. So it has like a double whammy. And this combination tends to amplify the building of bone and building of muscle. For older people, this is important. I mean, my father recently had was hit by a four-wheel drive as he was crossing the street, and he was thrown about 3 meters, not a fracture, let alone a broken bone. If it was a Vagoon, he would have shattered in multiple pieces. You know, it happens to them like Garth Davis, you know, the guy that loves goats, apparently, you know, that doctor, vegan doctor. And, you know, can you imagine being hit by four-wheel drive? And Dad's nearly 90. He'll be 89 in January, which isn't that far away. So he's got a bit over a year to get to 90. So hopefully nothing else runs him down like a donkey, a wild donkey from the old country. Yes, we don't want that. I've told you to look out for donkeys. So, and goats. In particular, ones that are actually reared by vegans. The thing is, Dad's been doing this for a decade, where he was becoming slightly sarcopenic. So there is hope for even you older folk out there that may have brittle, weak osteoporotic bones and want to biohack your way to stronger bones and all that. You got to take, I mean, Dad's on 6 grams of taurine, obviously. You know, it's a nice combination. 6 grams of leucine, 6 grams of taurine. Wonderful stuff. You know, amplify protein synthesis, amplify the effects of building stronger bones and muscle into old age. It's necessary. If you're going to preserve yourself and you're going to keep your muscle skeletal in a state where you're not going to be struggling. So as far as I'm concerned, you've got the information now, but I just wanted to cover those little points because you've got to understand the problems that epidemiology faces. Anyway, see yous.